Hello students, how are you? I hope you all are fine. Students, today we will continue our chapter 2, sexual reproduction in angiosperms. Yesterday we have discussed up to the fertilizations. Today we will discuss about post-fertilization changes. Means what happens after the fertilization. Soon after the act of the double fertilization, the flower begins to lose its shine. Means the petals, the stamens, the style, they fall off. The calyx may also fall off in some cases, but in some cases it may persist. For example, in tomato, brinjal. And the major events that act after fertilization are: number 1, development of embryo. Development of embryo. Second is development of endosperm. Third is development of seed from ovule. Fourth is development of fruit from ovary. We will discuss these events one by one. Firstly, we will discuss about the endosperm. And the development of endosperm. What is endosperm? Firstly, I will tell you about the endosperm. Endosperm is the food-laden tissue which is formed during the development of angiosperm seeds that provide essential nutrient to the growing embryo and also the young seedlings at the time of seed germinations. Now, based on the first and the subsequent divisions, endosperm is of 3 types. First is nuclear endosperm. In the nuclear type of endosperm development, the primary endosperm nucleus divides by repeatedly mitotic divisions. ठीक है जी. These nucleus divide by primary mitotic free divisions without the formation of wall. It results in the formation of a large number of free nuclei in the central cell of the embryo sac. Here the large number of nuclei are formed. A big central vacuole develops in the embryo sac, pushing all the embryos towards the periphery. The free nuclei, these free nuclei come to lie between the embryo sac as well as the vacuolar membrane. This is the vacuolar membrane, this is the embryo sac wall. These all lie in between this area. Okay? Now, from here the second phase begins with the cleavage of multinucleate peripheral cytoplasm. This peripheral cytoplasm, इसकी cleavage हो जाएगी. Cleavage means it starts developing wall around each central vacuole. ठीक है जी. Finally, all or most of the endosperm is converted into a cellular tissue by the formation of centripetal walls against all nucleus. ठीक है जी. This is the final nuclear endosperm. This is found in coconut. In coconut, the endosperm is multicellular, means these are number of cells in the outer part and free nuclear in the central part. Nuclear endosperm is the most common type of endosperm found in the angiosperms. And second one example is acyl indica. One example is coconut, second example is acyl indica. Now, second type of endosperm is cellular endosperm. In this cellular type of endosperm, the first nuclear division of the primary nucleus, it is followed by longitudinal or transverse. This is longitudinal, this is from this side it is transverse cell wall in the central wall. Subsequent nuclear divisions and wall formation. These wall formation result in the formation of cellular type of endosperm. ठीक है जी. At last, this is a final picture of cellular endosperm. This cellular endosperm is found in Petunia, Datura and Balsam. This is found in Petunia, Datura and balsam. Next one is third. It is halobial endosperm. It is halobial endosperm. In this type of endosperm, the primary endosperm nucleus moves towards the chalazial end. This is the chalazial end. In the structure of the ovule, we have studied about the chalazial and the micropylar end. It moves towards the chalazial end. The first nuclear division of the primary endosperm. This is the first nuclear division. It is followed by the subsequent division of the nuclear endosperm, which result in the formation of chalazial cell and a large micropylar cell. This is the small chalazial cell. This large is the micropylar cell. And at last, this is the chalazial, here chalazial, here chalazial. This large is the micropylar cell. ठीक है जी. At last, nucleus of the large micropylar cell divides. This cell divides by free nuclear division and further development takes place. This type of endosperm is found in all monocotyledons. This type of endosperm is found in all monocotyledon endosperms. Next topic is development of embryo. Students, we will study development of embryo in two parts. One in dicotyledons and second in monocotyledons. Yes, from here. In a typical dicotyledonous type of embryo development, firstly the zygote enlarges in size. This is the zygote. It enlarges in size and it secretes its own cellulose cell wall and divide by transverse divisions. Divide by transverse division and result in the formation of two unequal cells. The larger basal cell is the suspensor cell and this is the basal cell, or we can say it is the suspensor cell. Basal cell, suspensor cell, and the smaller one is known as the terminal embryo cell, which lies towards the chalazial end of the embryo sac. ठीक है जी. Next, the basal suspensor cell. The basal suspensor cell divides by transverse division and terminal embryonal cell divides by longitudinal division. These cells divided by longitudinal divisions and this divide by the transverse division to form a tetrad. To form a tetrad. 1, 2, 3, 4 tetrad. ठीक है जी. Out of the two cells formed by the division cells here. Out of the two cells formed by the transverse division of the suspensor cell, the lowermost cell towards the micropylar end enlarges in size and to form the vesicular basal cell. This is the vesicular basal cell. The other cell divides by variable number of transverse divisions to form the longer suspensor cell. Here it is longer suspensor cell. It consists of 6 to 19 number of cells. Each of the two cells at the terminal, it divides longitudinally to form 4 tetrads. ये दोनों longitudinally way के साथ divide करेंगे और when they are in two number we call it two cell proembryo. तो when they divide they form 4 cells. ठीक है जी. They form 4 cells and this stage is known as the quadrant stage. This stage is known as the quadrant stage. Each of the 4 cells divide by transverse division to form 8-celled octant stage. To form 8-cell octant stage. Then the 8 protodermal cells. These 8 protodermal cells, 4 are visible, 4 are at their back side. ठीक है, these protodermal cells divide by pericranial division to form an outer protodermal cell and an inner cell. This is the outer protodermal cell and these are the inner protodermal cells. 4 protodermal cells, 4 outer protodermal cells. ठीक है जी. Then the 8 protodermal cells divide by anticlinal divisions. These divide by anticlinal division to form the embryonic surface of the epidermis. Here these cells divide by anticlinal division to form the epidermis. This black I have shown you about the epidermis. ठीक है जी. The 8 protodermal cells divide हो गया. Then the 8 inner cells. These 8 inner cells divide in different planes to form the ground meristem. This is the ground meristem of cotyledon. This is a cotyledon. This is the ground meristem. That shows in red is known as the ground meristem. Now the suspensor cell situated at the tip. Here the suspensor cell situated at the tip of the, it is known as hypophysis. This is known as hypophysis. This cell is known as the hypophysis. Now this hypophysis again divide by transverse and longitudinal divisions to form 3 tier of cells. Here 1, 2, 3. 3 tiers of cells. The cells of inner tier towards the embryo form the radicle. Inner cells form the radicle and the cells of the middle and the outer tier forms the root periderm as well as the root cap. Here it is the root cap. ठीक है जी. At this stage the developing embryo is globular and shows radial symmetry. It is called the proembryo. Here it is called the proembryo. 2 cotyledons arise from the 2 ridges of the tissues appeared at the distal region of the proembryo and radicle is formed and a radicle is formed and the cotyledon grow rapidly, whereas a few cells become the basis of the cotyledons to differentiate a dome-shaped plumule. This is the dome-shaped plumule. This is radicle, this is dome-shaped plumule. These two are the cotyledons. Hanji students, next here is the development of monocotyledonous seeds. Early phases of embryogenesis in monocotyledonous seed up to the formation of globular embryo is very similar. यहाँ तक सारी stages monocotyledon की और dicotyledon की same है. ठीक है. In monocotyledons about 1/2 of the terminal cell derivatives. Here 1/2 of the terminal cells they function as root cell. ठीक है जी. And stem cell. These cells function as stem cell. This is suspensor and here is the plumule. Stem cells and plumule. Plumule, hypocotyle, root. ठीक है जी. These 3 cells are formed in this stage up to A, B, C. These stages, these all are same just as in the dicotyledons. ठीक है जी. In monocotyledons, about 1/2 of the terminal cell derivatives function as shoot apex, whereas the remainder cells show rapid rate of cell division in the formation of single terminal cotyledon. Single terminal cotyledon. This is the embryo of the monocotyledon: suspensor cell, hypocotyle, and stem. Next event is development of seed. Students, double fertilization in angiosperms result in the formation of ovule in the seed. Means the zygote develop into an embryo and the triploid primary endosperm nucleus, it gives rise to a nutritive medium that is known as the endosperm. ठीक है जी. एंडोस्पर्म में persist और completely digested during embryogenesis. कई बार एंडोस्पर्म यूज भी हो जाता है पूरा तो वो ड्राई फ्रूट्स बन जाते हैं. कई बार वो यूज नहीं होता है तो वो फ्लैशी फ्रूट्स बन जाते हैं. ठीक है जी. तो जिनमें ये यूज हो जाता है दे आर नोन एज एल्ब्यूमिनस फ्रूट्स, फॉर एग्जाम्पल व्हीट, कॉर्न, ओनियन एंड पाम. इन विच इट मे परसिस्ट दे आर नोन एज एक्स एल्ब्यूमिनस फीड्स. ठीक है जी. नेक्स्ट, समटाइम्स albumin may remain as a reserve food material in the seed. हाँ जी. Now the nucellus is generally used up. Nucellus तो use हो ही जाता है during the development of the embryo, but in some cases it may remain persist, and that is known as the perisperm. Now the outer integument becomes hard. Outer layer hard हो जाती है, leathery हो जाती है. That is known as the testa. ठीक है जी. This testa ensures the survival of the seed. The inner integument may persist. Sometimes this red inner integument may persist, or sometimes it may used. It is known as the tegmen. It is known as the tegmen. This red is known as the tegmen. This is known as the tegmen. The inner integument if persists, it is known as the tegmen. Now the microphylla remains in the form of fine pore on the surface of the seed. Here fine pore, that is known as the microphylla end of the seed. Funicle is transformed into the stalk of the seed. Funicle वो seed की डंडी बन जाती है. And the hilum marks the point of the attachment to the stalk. ठीक है. The seed, this is the cotyledonous node, plumule, radicle. If we open the seed, it looks like this. ठीक है. The seeds of most of the plants germinate when they got the favorable conditions. ठीक है. However, many plants whose seeds although viable but they do not germinate after getting even the favorable conditions. Then germination of some seeds may be delayed for days, for a week, for a month, or for years. That condition is known as the dormancy. That condition is known as the dormancy, when seed is not able to germinate even after getting the suitable conditions. This is enough for today, students. We will discuss about the development of fruit in the next session. Thank you and have a nice day.